the great pleasure of welcoming you all to this evening's expert lecture, one in a series of expert lectures that have become a feature of the intellectual life of the university. And so thank you all very much for attending this evening. And now I would like to ask the Vice-Chancellor and Principal, Professor de la Rey, to come and welcome you and also to introduce the speaker to you. Thank you.
Good evening, ladies and gentlemen, and thank you very much to Professor Robin Crew for standing in for the Dean, but also for his gender sensitivity. Professor Crew, may I offer Program Director for future reference? Thank you, Professor. Ladies and gentlemen, it's my pleasure to also welcome you to this 18th lecture in the UP Expert Lecture Series. And I'm delighted to welcome a number of representatives of government, industry, a number of the UP alumni who I see in the audience, my executive colleagues, colleagues from across the university, and many of our students. It's a special pleasure to see our students in the audience. As I indicated, ladies and gentlemen, this is the 18th in the university's Expert Lecture Series, which we launched in 2010. The purpose of our series is to provide a public platform for some of our academic experts at the university to share their expertise with a general audience, and particularly to inform us of some significant developments in their particular field of expertise, with a focus to developments that may have an impact in the future. The lecture series was introduced very shortly after my appointment as Vice-Chancellor and Principal, and looking back over the 18 of these, I do believe that there's ample evidence to show that the series has been very successful in showcasing the expertise here at the university across our various faculties. I do consider it to be an important element of our general outreach program, making available to our broader community the assets that we have, both knowledge assets and other assets of the university. And it's one of the ways in which we seek to make a meaningful contribution to the development of our society, but in particular in sensitizing the general public to the importance of science in relation to the country's economic and social development. Over the series, we've covered a number of topics, ranging from issues such as human rights. I remember Professor Michael Pepper's lecture on stem cells. We've covered topics such as leadership, the digital highway, energy efficiency, and tree health. As I look up, I'm reminded of the occasion that Professor Mike Wingfield presented his lecture. But I think it's also fortuitous this evening, as I look back on the success of the series, to recall that the very first lecture was presented by Professor Robin Crew, when we started it on the evening of the 26th of August 2010. On that occasion, I have the full list in front of me in case you're wondering. Professor Crew's title was Doomsday Scenarios and the Fate of the African Honeybee Population. And he spoke to us about the large-scale loss of honeybee colonies posing a threat to honeybee populations then in the US, Latin America, and Europe. And at that time, it featured quite prominently in the media. While the problems facing that part of the world seem to have gained ascendancy locally, because just last week, when we hosted the NRF Public Lecture Series, which was delivered by Professors Slippers and Myberg, after a lecture which was broadcast via SAFM, there was an opportunity for the lecturers to phone in. And I recall on that occasion, about the fifth caller asked the question of honeybees, Professor Crew. And I think Professor Slippers answered it quite ably on that occasion. So I was reminded and wondered how you would have dealt with it. But I have read recently that this is affecting us quite seriously, and particularly in the Western Cape. And I mention this and go back to the very first because it does amplify the point that I made earlier, that what we're looking at or asking our expert to inform us of is the developments in their field, and particularly to suggest any large-scale implications that it may have for all of us. And I think tonight we will not be disappointed in that regard. So without further ado, I'm delighted to introduce to you our expert lecturer this evening, which is going to be presented by Professor Millar. I did get his CV, and I must tell you it was quite a challenge to keep this introduction down to manageable proportions. So I have been highly selective. But let me start off by saying Professor Millar was born in South Africa. He grew up in Zimbabwe, where he did his first degree, and then he studied in the UK. He then took up a position in South Africa at the University of Cape Town, and then later at the University of Edinburgh. He returned to South Africa in 2011 to take up the position of Director of the Mammal Research Institute here at the University of Pretoria, with a secondary position of Director at the UCT MRC Reception Biology Group, having led a review of that group a bit earlier. He occupies a number of positions in recognition of his outstanding academic career. So let me mention a few. He is also a research fellow currently at the Centre of Integrative Physiology at the University of Edinburgh, and an emeritus professor at the University of St. Andrews. When he was at Edinburgh, he was director of the MRC Human Reproductive Sciences Unit, which consisted of close to 100 researchers. And it's one of the discussions that I have many times in the university, question of what makes a viable center or an institute in a university. 100 researchers, I think, is quite outstanding. While he was there, he secured a significant amount of funding during his tenure. I think it was in the order of about £70 million. And at that time, his research focused on pathologies of female and male reproductive tissues, infertility, contraception, and hormone replacement therapy. He was founder and chief scientific officer of the very successful reproductive health company, Ardana Biosciences, which raised £73 million and was listed on the stock exchange, the London Stock Exchange. And it developed a number of drugs for the market. Professor Millar, could you do something for our university while I mention this? Professor Millar has, in summary, interacted with a number of major pharmaceutical companies. He's been on the board of quite a few, and he's held visiting professorships at so many universities that I don't think I should mention all of them. If I just move on very quickly, he has received 4 successive A ratings by the National Research Foundation of South Africa. And just to remind us all, an A rating means that you have been judged internationally by your peers as pioneering and leading in your respective field. As a clinical chemist, he's had approximately 30 years' experience in basic and clinical neuroendocrinology. Professor Millar has published over 400 papers. He holds 18 patents. He has an H index, for those of us who know what this means, of over 60. And his work has been cited more than 17,000 times. He has served on the board of many international journals and is currently editor-in-chief of Neuroendocrinology. As I said, I think there are too many boards for me to mention to you this evening, but I do wish to draw your attention to the information that's available in front of you on the desk, and if you don't have it, please let us know, we'll make it available. But very recently, he was elected president of the International Neuroendocrinology Federation for a 4-year term which will expire in the year 2020. Professor Millar has also received a number of medals and awards, among these the Wellcome Gold Medal, the Oppenheimer Gold Medal, the Jeffrey Harris Laureate of the International Federation of Neuroendocrinology, and the list goes on. As I'm sure you've gathered, Professor Millar's research focuses on the neuroendocrine regulation of reproduction, and he will share more about that this evening. For now, it's my distinct honor to invite the distinguished Professor Millar to address us on the topic, which is intriguing, A Hitchhiker's Guide to the Galaxy of Brain-Body Dialogues. And I hope all of us recognize the title and to which it refers, at least those of us of a certain age group. Professor Millar.
Thank you, Professor Delaray, for the over-generous introduction. I'd like to turn things upside down and first acknowledge the people who've helped me in my scientific career, instead of leaving it to the end, where often I run out of time and then forget to do so. So first of all, thank you all for attending, and I hope that you'll enjoy the ride and the Hitchhiker's Guide to the Galaxy of Body and Brain Interactions. So I'd like to thank the University of Pretoria, who've been very, very supportive of me in the last 4 years, Professor Delaray, Professor Struer, Professor Wingfield, Professor Lubuma, and Professor Chimimba, as well as Simon Kotzer and his wonderful team of assistants at NAS. And then Professor Buch and Professor Pepper, who's kindly attended and has helped us in establishing the Center for Neuroendocrinology in the Faculty of Health Sciences, and as well as Professor De Jaar. And there are many others, and particularly the support staff, who have been really kind and helpful to me during my 4-year tenure here. Then I'd like to thank the many, many students and postdocs that I've had over the years and had the opportunity to mentor them and to have such fun in asking research questions. I mean, there is nothing nicer than the dynamic of sitting down with bright students and hearing their ideas and sharing your ideas. And that's what makes me an academic. And I think when I retire, that is going to be one of the things I miss most of all. I also, and in fact, we're hoping to have new students now with this new Center of Neuroendocrinology at the Faculty of Health Sciences. So any of you who'd like to work in this area, then you can always contact me. And lastly, the agencies who've supported the research, the National Research Foundation, the MRC of both the UK and South Africa, National Institutes of Health, National Science Foundation, Cancer, and a lot of smaller agencies. Without that, it would not be possible. So we can get on with the talk now, and I just need to arrange a few things. So this is the inspiration from the book, and I recommend that you read that book. The answer to the story is 42, some of you will know. But don't panic is the first message from the book. I will not be presenting a lot of data. I'll have a lot of pictures and a lot of conclusions and some headlines, but I'll have very little data. So I hope that'll make you feel more relaxed. Now, we live in a changing world. This shows air temperatures during the year. And even at the equator, you'll see that there are considerable changes, and even photoperiod changes at the equator. And all living organisms have had to adapt to be able to survive in a world of change. And seasonal sexual reproduction is one of the most impressive. Now, Aristotle, from 300 odd BC, was the first recorded person who noted these breeding cycles and noted how animals interacted with their environment. He was an absolutely astonishing man because he wrote this book. This is a copy from Constantinople, which is a history of animals. And in that, he recorded every single animal that he knew of on the island of Lesbos and then went further afield. And this, in 10 parts, is a most extraordinary treatise. It remained the reference book in biology for 2,000 years, until the 16th century, when new biologists were spawned. And the accuracy of his observations was so extraordinary. For example, he noted that in the squid there was a special tentacle that was for sexual reproduction. What it did with that tentacle was to remove a purse that contained spermatozoa and insert them into the female. And that was never re-explored until the 1960s, when it was established that's exactly the way that they achieved fertilization. Now, he trained under Plato for 19 years, and that was in philosophy. And the two questions in Greek were the what and the why, and he applied that to all of his biological studies. Now, although he has enormous accuracy in his recording the what, the why is sometimes wrong. So I'll give you an example. Aristotle observed, and people knew that if you cut off the testicles of a bull, he was no longer fertile. But for 4 days, while the tube still hung outside of the abdomen, the bull could cover a female and she would become pregnant. So his interpretation was that the testes were simply weights to keep these tubes, sperm tubes, out of the body. So his observation was perfect, but his interpretation was flawed. So this is the message. Association does not equal causality. And that is a scientific mistake that is made so often in our research, and actually in the genomic era even more. So this association does not equal causality, and I hope that we can remember that lesson. So what my talk today is how the external environment impinges on the brain and gives it messages, how it influences the body, and how the body, its internal environment, talks to the brain. And all of this is integrated by the brain to influence body function. And Geoffrey Harris at Oxford was really the father of understanding the way in which the brain could communicate to the body. What Harris did was observe that between the base of the brain, called the hypothalamus, and the pituitary was a collection of vessels that appeared to be capillaries. And this is only a couple of millimeters. So he injected Indian ink and showed indeed these were capillaries connecting the brain to the pituitary gland. And he then set out to try and find out what these factors might be. So in essence, this is what neuroendocrinology is. It's the way in which internal environments, external environments are perceived by the brain, integrated in this area, the hypothalamus, and conducted in vessels of hormone secretion to the regulation of the body and the brain. Now, this is a busy slide, but if you'll bear with me, I'd like to spend a little time going through it. So this really is the hypothalamus, the base of the brain, and here are a whole load of neuropeptides that have been isolated. And each of them regulates a certain pituitary function. So CRF stimulates ACTH, which stimulates the adrenal to produce cortisol and all the effects of that. TRH stimulates TSH, the thyroid gland, and metabolism. Growth hormone, which targets the liver and bone and the muscle. Prolactin, stimulating breast development. And then my favorite area of the gonadotropins, FSH and LH, that stimulate the ovary and testis. Then the posterior pituitary produces a thing called antidiuretic hormone, which regulates water homeostasis in the kidneys, and produces oxytocin, which stimulates the uterus, the breast, and also smooth muscles in the ductus deferens of man. So what I'm going to talk about today is something that oxytocin does in terms of behaviors rather than these targets. Because these structures, molecules, can be used for many different purposes and get recruited during evolution for that purpose. Then I'm going to talk about these gonadotropins and the regulation of the testis and ovary, and particularly on sexual differentiation and how female and male brains are quite different in lots of ways. That's daring, isn't it? And then I'm going to speak about the stress pathway regarding the adrenal, and I'm not going to cover the other areas, too vast. It'll be the whole galaxy. But there is another area that's not mentioned here, and that within the hypothalamus is an area called the arcuate nucleus, and that harbors the neuropeptides that are involved in appetite regulation. And as we'll hear later, we have a pandemic of obesity, and that's a very important area. So if we return to the reproductive system, this hormone, gonadotropin-releasing hormone, stimulates pituitary, testis, and ovary, etc. And we didn't know that structure. And this man, who I had the fortune to work in his laboratory, Andrew Shalley, received the Nobel Prize for isolating that hormone from about 1.5 million pig brains, of which he got about 1 milligram. It was a tour de force. And this was the structure. It's a simple peptide, 10 amino acids. And that regulates your entire reproductive system. Less than 1,000 neurons are responsible for producing this in the brain, and that is the whole kernel of reproduction. But Shelley claimed that this structure was exactly the same in all vertebrates. And I had reason to not agree with this. I could not see how that would be the case. So with Judy King, her MSc student in Cape Town, she showed that yes, the mammalian form was present in amphibians, but there were different forms in terms of their charge in birds, reptiles, and fish. And Judy then decided to go on and actually show what that structure was. So she ended up with the tour de force of collecting 600,000 chicken brains, from which she purified 11 millionths of a gram, which was sufficient for her to work out what the structure was. And the structure was, this is only the second that was ever purified. Here's Shelley's molecule, and here's Judy's chicken molecule. And that set the scene to actually work out the structures of many of these, covering all vertebrates and the progenitors of vertebrates, the chordates. And what you'll see immediately is the blue areas are the amino acids that are completely conserved, representing 600 million years of evolution. So we believe that these experiments of nature tell you a lot. You don't have to do structure activity. Nature's already done it. So we felt this N-terminus and C-terminus engage with the receptor, and therefore this must be a folded molecule. And with a lot of different approaches and so on and so forth, we can confirm that, and we showed this is the essence of the molecule. The C-terminus and the N-terminus bind the receptor, and this part does not, but this here flexes the molecule to allow it to bend in this way. And if you substitute that amino acid with one that forces this conformation, you increase the activity of the molecule over 100-fold. And that's the nature of all agonists of this molecule. But this N-terminus is responsible for activation of the receptor, so therefore substitutions here create antagonists of the molecule. And these have become wonderful tools for treatment of disease. Now, all that was done in the absence of knowing anything about the receptor, and it became important to work out what the receptor was. And with Stuart Sealfon's lab in Sinai, we cloned this first mammalian GnRH receptor. Now, what it is, this is a membrane of the cell, and this receptor snakes in and out of the membrane like this, and these are called transmembrane domains. And the hormone will bind on the outside and send a signal on the inside. And I'm not going to go into that in any detail. But by doing mutations of nearly all of these amino acids, or a very large number, we figured out that these red ones are the ones that interact with the hormone directly. And we assembled models based on other G protein coupled receptors, etc. And to make a long story short, these are these transmembrane domains as spirals in the plane of the page here. These were the residues we identified as being the ones that interact with the hormone, and we could satisfactorily engage the hormone and accommodate the interactions through that model. So this was very instructive in planning our thoughts about how to develop other molecules. And in fact, the new generation of GnRH small molecules is entirely dependent on that. But just to summarize, these are all the GnRH molecules that are used in the clinic today. Here is the substitution of the glycine to force the folding in these agonists. Then in the antagonists include that, but modifications of the N-terminus so it cannot activate the receptor and competes with the endogenous hormone. So both of them can be used nevertheless to inhibit this activation of the pituitary. The agonists by desensitization and the antagonists by competition with the endogenous ligand. So very briefly, these are the clinical applications of these GnRH analogs. The GnRH itself can be delivered in a pulsatile way, in the same way that the brain does, in order to be pro-fertility, stimulate reproduction. On the other hand, agonists and antagonists can be used in the anti-fertility mode, or they can be used in an anti-cancer mode. So, for example, GnRH analogs are the biggest treatment for prostatic cancer. And then there are a whole load of women's health conditions in which they've also been used. So, the first thing is this diversity of GnRH structures evolved over 600 million years. So we thought that we should try and work out the phylogeny and relationship. So now we're looking at the genes. This is some way down the line from the original work. And they clustered into a GnRH1 family, a GnRH2 family, and a GnRH3 family. And this has been silenced in anything above amphibians and teleos fish. But these two exist in mammals. So this one we knew was to stimulate the reproductive system. This one, we didn't know what it was for. But the astonishing thing about GnRH2, which you can see represented as chicken 2 here, is that this beastie, this molecule, is completely conserved from fish all the way to man. So we figured that must be pretty important. What could the function be of such a molecule? The distribution of GnRH2 in the brain mapped onto areas associated with reproductive behaviors. So we thought this was a likely candidate and proposed that the GnRH2 was a neuromodulator affecting sexual behavior. And did a lot of experiments. Here we stimulated green iguanids to display sexual behavior. Here in ringed doves, 20 minutes after administering, they go through this courting behavior and mating. And then in female song sparrows, you can see the solicitation of males is increased by GnRH2, but not the GnRH1. And the musk-shrew, which is a beautiful model of sexual behaviors, with Emily Rissman, we could show that the number of female receiving mounts is markedly increased by treatment with GnRH2. But the strange thing was that would only work in the shrews if they were deprived of food. If they were fully fed, we could not increase the amount of sexual behaviors. So it seemed to me that obviously the system was going flat out if they were well fed. If you're well fed, you should reproduce. If you're badly fed, you shouldn't reproduce. So we decided to look at the effects on feeding. And this is what we found. There's a 30% reduction in the feeding of these shrews when they're given GnRH2. So we worked out a kind of model that when there's good food intake, it stimulates GnRH2, which stimulates sexual behaviors, mounting, etc., etc. At the same time, the increased GnRH2 feeds back on the brain to inhibit food intake. So there's this very close relationship of food and sex. Some of you know about the chocolate story. Okay. So we asked the question, does GnRH2 have similar effects in humans And can GnRH2 analog stimulate libido and reduce appetite? In other words, it would be quite an attractive therapeutic. So we did this in marmoset monkeys, females, and this is what's called proceptive behavior. In other words, they're recruiting the male. They're going to the male and say, hey, how about it? And you'll see that with increasing doses of GnRH2, we could increase this behavior, but GnRH1 had no effect. So this elicited a lot of interest in the scientific world, but also in the media. And you'll see here, this was an article we published in, I think we interviewed in Scotland on Sunday, and the next thing it would go on viral, and here's a BBC thing saying, hope for sex boosts slimming pill. And they remarked on the fact that around 40% of women in the Western world are said to experience low libido at some point or another. So, to quickly summarize this section, I showed you that GnRH produced in neurons in the hypothalamus is secreted into portal vessels to stimulate the pituitary gland. This is a neuroendocrine way of operation. But GnRHs can act as neurotransmitters for that behavioral way that I told you about. And then there are other systems where GnRH produced in a cell can influence an adjacent cell. This is called paracrine. And then in tumor cells, we demonstrated secretion of, and other people, production of GnRH and expression of GnRH receptors, suggesting there could be an autocrine regulation. So this is an example of the testis. Very quickly, this is a transgenic mouse whereby the promoter of GnRH drives green fluorescent proteins. So we see here in the Leydig cells that GnRH is being expressed, and that we know that the Leydig cells communicate with the Sertoli cells and spermatogenic cells in regulating spermatogenesis. And Colleen Flanagan, who's in the audience, I think, and myself and Karen started to look at the possibility that GnRH receptors were expressed in human breast carcinomas. And so we published this article in Science and then went on to actually show with cell lines that you can inhibit tumor proliferation with analogs of this hormone. And I'll just show you a related example that in mice, nude mice, that tumors in the control are much larger than tumors on one of these GnRH analogs we made. And that's quite impressive data because here's the growth of tumor without treatment, and here's the growth of the tumor with a GnRH analog which we designed, whereas the GnRH antagonist seems to enhance disease. So this comes to the end of this particular part of the talk, and what I'd like to try and emphasize is that the hypothalamic pituitary gonadal axis, by delineating that, we've opened up many, many important things in human existence. The first is the development of the contraceptive pill, which is arguably the single most important contribution to women's liberation. The second is the development of hormone replacement therapy. And the number of women who have not had to suffer loss of bone, not had to suffer complicated cardiovascular aspects, and cognition, which is influenced enormously by estrogen, is pretty substantial. And then this system has been used for the generation of a lot of treatments for breast, prostatic cancer, and other hormone-dependent cancers, both in the form of antagonists of steroids or enzymes which will not allow their elaboration, or simply GnRH analogs. The sad thing is that with all these interventive discoveries, that much of it does not be taken up by people, and in fact the modification of behavior and lifestyle is often more effective than devices and the therapeutic drugs for some areas of human health. So obesity is a classic example. Obesity is a dreadful pandemic. It should be easy not to become obese, but we seem incapable of it, and the pharmaceutical industry has not managed to produce effective drugs. Sexually transmitted diseases, and contraception. I indicated that we have wonderful contraceptives now. Sexually transmitted diseases would not be a problem if people adhered to the use of protective sex, etc. So it's a really sad thing that for every advance we make pharmaceutically, there are potential problems because of human behavior. And here I think is a really lovely picture of one from Rudy van Arda, who's in the audience here. This here is a football that's being sold by a Malawian boy, completely constructed from thousands of condoms. And that is because the free condoms are available, but they're not being used. So he's become very enterprising and starts to sell footballs. Okay, so I'd like now to mention another experiment of nature. And this involves inactivating mutations of neuroendocrine pathways that are stimulating the advance through puberty and adult reproduction. And my group is fortunate to be part of a large European consortium. In fact, we're the only member outside of Europe where the whole genome sequencing is being done on patients who fail to progress through puberty. And this has revealed some really exciting new neuroendocrine pathways that regulate the GnRH system. So, these are some of the genes that have come up. So this is the same cascade going down to the gonads from the brain, pituitary, etc. These are the different levels at which things can go wrong. And in the blue are the genes in which mutations have been found which have caused the condition. And I'm only going to give you one example. There are lots that I could show you, and some very exciting ones. This is simply an inactivating mutation of GnRH1, which you'd expect to give the results. And these are two siblings, 18-year-old brother, 17-year-old sister, who have the homozygotes. And you can see that they have not advanced through puberty at all. And having identified what the lesion is, you can then deliver GnRH in pulses in a subcutaneous way. And this is the sort of result you get. That's the normal adult male with pulses of GnRH, which are represented by pulses of LH. Whereas the GnRH deficient male, you see nothing. And if you give GnRH in a pulsatile administration to each of these pulses, you see a very nice LH response, bringing it back into the normal range. And these girls begin to ovulate, and the men start to have testicular development, etc. So, okay, enough of that. So I'm just going to mention a very brief aside, okay? And this is on an area called optogenetics, which is the most fantastic development in modern science, especially for neuroscience, because the brain is so complex that it's very difficult to look at individual pathways in a living animal. What optogenetics does is you take the promoter representing the gene in a particular nerve cell and connect that up to a protein called channel rhodopsin. Rhodopsin is the light pigment that recognizes light. You then put that into an adenovirus and deliver it to a part of the brain, but it'll only express in those neurons which have the machinery to recognize the promoter. So you now have this channel rhodopsin, which will respond to light being shone in the living animal, but will only activate those neural pathways which you have targeted. So you can start to see what the outcome of that is. And in the GnRH system, this is Alan Herbertson's work, you'll see here that when he's shone blue light on these living rats, he can get a peak of LH representing a peak of GnRH. And so this is really opening up amazing possibilities for looking at the function of the brain. So just to summarize what's been identified, this is the GnRH neuron. We didn't know about any of these here, but these have all been revealed because of inactivating mutations that were identified in patients who failed to go through puberty. So you can imagine what a rich field these experiments of nature are revealing to us. So these mutations in a failure to advance through puberty. They provide informative counseling and therapeutic options for the patients. They also reveal these novel pathways, which I've talked about, and they then become new drug targets. And indeed, we've made and developed antagonists of some of these particular neuropeptides. So I want to move on now to an area which is called brain sexual differences or dimorphisms. Why the male and female brain are different, and how different are they? And also sexual differentiation during development. So now Aristotle, to return to my hero of the history of animals, his thoughts on sexual differences were, and I'll read it out, in all genera, nature makes a differentiation into the mental characteristics of the two sexes. This differentiation is the most obvious in the case of humankind. I wouldn't say that is necessarily so. With all animals, the female, and this, I was going to have a photo of Cheryl come up here. The female is softer in disposition than the male, is more mischievous, less simple, more impulsive, and more attentive to the nurture of young. The male, on the other hand, is more spirited than the female, more savage, more simple, and less cunning. And I do think this is the case. The traces of these differentiated characteristics are more or less visible everywhere, but especially visible in man. So let's see if it's true. So Baer and colleagues represented this idea of man's simplicity by a single switch, on and off. The woman, on the other hand, has an on and off switch, but also has this array of regulatory things, so much more cunning and much more calculating. But what are the facts? Well, if you look at the size of structures in the brain, these pink ones are structures that are larger in healthy female brain than the male. And the blue ones are ones that are larger in the male than the female. And there's substructures which have great sexual dimorphism. So there is very little doubt, and I mean, it's a reputable journal, so you better go buy it. So what about these effects of performance in the brain? So there's a thing called a Morris water maze. You make a rat swim around, and there's a little platform hidden under the water. And it takes time for them to learn, but eventually from the start, they'll go directly to the platform. Now, how do females and males do in this? The males seem pretty constant. This is a latency time. The females do really badly when they're in estrus, but they become much better when they're at the other part of the cycle. So no premier should be a female and make decisions at the time of estrus. Now, here's an amusing little add-on that Geoffrey Millar looked at, and these are lap dancers and what their earnings are. And these are women who are just ovulating and not on the contraceptive, and these are the ones on the contraceptive pill. And you can see there's a really good connection as to the amount of the tips that they get according to whether they're ovulating or not. So the men might be very simple, but I think they can detect certain things. Okay, in chimps, there are beautiful examples. For example, fishing for termites. The females do much better than the males. This is the age at which they learn to do this. Look at the old males, way over here to the right. They're just about geriatric before they learn. Okay. But there are tasks which favor men, and these are various types of tasks. These are the ones men do better in. Mental rotation of 2D images of a 3D object. Mental unfolding. Target-directed motor skills. They learned that in the pub. And then the disembedding. Men are very strong about that, being able to see this image. And at mathematical reasoning. On the other hand, women are much better at mathematical calculation. I'd be interested to hear what Roman thinks about this. And the memory for object displacement, for manual precision tasks, for a fluency of this type, and perceptual speed, noticing there's no chimney there, and noticing there are no bars there. Who saw that first? I should have said hands up. Okay, so the women and men are very different in their brain functions. There are lots of areas, of course, that are very similar. So let me give a couple of examples of really persuasive ones. The rates of serotonin synthesis can be studied by PET analysis. Now, these are the men. That's the amount of serotonin at basal levels, and these are the females. Quite clearly different. And if you deprive them of the precursor of serotonin, tryptophan, the men don't change, but the females have changed very significantly. So these results are important because they may help understand why some disorders, such as unipolar depression, that involve serotonin dysfunction, do not equally affect men and women. So this business of finding out about different operations of the brain is useful in terms of understanding about therapeutic approaches. So the implications, many CNS disorders show sexual differences in their incidences or severity, and it includes a lot of things like Alzheimer's, post-traumatic stress, anxiety disorders, etc. So we feel that that will be very useful in going forward on this. Now here's an example, for example, in addiction. So these are scans. If you look at women, when they're not on cocaine, the amygdala lights up. When they go onto cocaine, the amygdala goes down. Men are exactly the opposite. So even in things such as addiction, there are strong differences in the brain which need to be taken into account in a therapeutic sense. So how do these sexual differences or dimorphisms of the brain come about? There have been many experiments to demonstrate this sort of thing. If you take a young pup developing a rat, you remove the testis of the male, and then this means his brain does not get programmed with testosterone during a window, a window of development. If you then give him estrogen as an adult, he has female sexual behavior and presents himself for mating. If you take a male pup and you leave the testis in place, in other words, it can program the brain, lo and behold, as an adult, when you give estrogen, no effect. What happens in females? Here's a female pup. She's been given testosterone in the important time of brain imprinting. If you inject testosterone as an adult, she goes in for male behavior. A female injected with placebo, you then give testosterone later, no effect, because the brain was not programmed at the formative time. So which parts of the brain are affected? And which genes are up or down regulated? And there's a huge body of work on this. But I'll only present one little snippet. And this comes from Margaret McCarthy's work. She shows that the male hippocampus uniquely starts to express a prostaglandin E synthase compared with the females when it gets exposed to estrogen. And this estrogen is rarely because testosterone comes to the brain, which is converted to the female hormone estrogen to impinge upon the male brain. So there's a lot of work about that. And the bottom line is that you might expect that there'd be lots of genes which get turned on or turned off as part of this sexual differentiation in the brain. But strangely enough, it's not very many, and that was just one example. So I now want to refer to another experiment of nature which tells you about the male and female brain. And this was something I became fascinated about many years ago. It's a condition where testosterone can't be converted to dihydrotestosterone, and dihydrotestosterone is the one that causes virilization of the external genitalia. So in this case, this is an XY, in other words, male child, but presenting with female external genitalia because of the lack of this enzyme. But the brain has been bathed in testosterone, so the brain will be a male brain. And lo and behold, at puberty, suddenly this guy grows a substantial penis and is fertile. And you can see he's become muscular. He doesn't have beard growth because that requires this. And the remarkable thing is the socio-psychological transition that happens here. So although this guy was brought up as a little girl, when he reaches puberty, he does the transition perfectly well. And there are some lovely stories about an Italian who presented himself at Mount Sinai because he was 50 years of age and he'd been acting out this female role in his Italian community for 58 years, hoping his mother would die so he could really now become a male. And, so they sort of proceed along those lines. But that tells you the power of how the brain is transformed. And now very quickly, if you look at the bed nucleus of the stria terminalis, you'll see it's a much larger feature in the male than the female. These are humans. In homosexual males, it's exactly the same as in heterosexual males. But in transsexual males, you see there's a sort of intermediate type size of the VNST. And these data are represented here. So, and now I want to very quickly go on to maternal and pair bonding and imprinting. So we know that oxytocin promotes uterine contraction for labor and milk ejection during breastfeeding. And the new aspect is that it's thought to influence nurturing and bonding. So in the hypothalamus, these neurons produce oxytocin, they release them, they target the uterus and the breast. And the uterus and the breast have positive feedbacks to activate these neurons further. So if you remove oxytocin during development, you fail to let this have a rise in oxytocin. You get in there, you do a Caesar, and you remove the lamb. She never sees oxytocin. She does not imprint on her lamb at all. She does not recognize the sound of the lamb, and she does not recognize the appearance of the lamb, which they always normally do. And you can overcome that by supplying oxytocin. So oxytocin is elevated in labor and in breastfeeding. So I just wanted to be provocative and study this and look at some publications. Do cesarean deliveries and the absence of breastfeeding affect maternal-child bonding in humans? Because I think that would be quite important. So there are a number of papers which support this. And here's just one example that the maternal brain response to their own baby cry, which they're measuring by MRI or whatever, is affected by cesarean section, supporting that notion. And then a Warwick Child Group has looked at breastfeeding and concludes that breastfeeding does help mothers bond with babies because well, this is now a popular thing that's come out. It's not the original paper. It releases the love hormone, which is oxytocin's now being dubbed. So what oxytocin does establishes this bond between mother and offspring. But we might ask that where there's a bond between a male and a female, does oxytocin play a role in that? So in other words, during evolution, if there's an existing hormone which can alter behavior, it can be used for another behavior. And we see that recurring theme over and over again. So now this is a very beautiful study, which is quite a lot has been done now, the diversity of social, sexual, and parental behaviors in voles in the Americas. There are two species that we're talking about, prairie and meadow vole. These guys are almost identical in their genome, very slightly different. But the prairie vole is highly social, monogamous, and biparental. The meadow vole is solitary, exactly the opposite, promiscuous and uniparental. So completely different behavior. And if you look in their brains and you look for oxytocin receptors, you see here in the nucleus accumbens a high expression in the prairie vole and nothing in the meadow vole. And if you correct that by giving oxytocin to this miserable old meadow vole, he turns into a lovely, companionable vole. And you can do that also by using adenovirus and deliver the gene into the right part of the brain, etc., etc. So I think these studies have been really interesting in some rodent species and other ones as well. But the question, I suppose, is does oxytocin have similar roles in humans? And I guess the jury's out on that, but there are some interesting things. So, for example, they looked at the expression of oxytocin receptor using PET scanning in women presented with maternal and romantic love situations, seeing their partner or seeing the baby. And different parts, red, romantic love, yellow, maternal love, light up in the brain. So there are different parts of the brain where oxytocin receptors are expressed in relation to these kind of inputs. And this is an intriguing paper where they looked at oxytocin in the cerebrospinal fluid in women with histories of child abuse. And here's the degree of child abuse, showing that there's quite a marked relationship of a decrease in oxytocin. And this only happens with emotional abuse, not with other forms of abuse. A Norwegian group has done a vast study of over 2,000 twins where they've looked at the oxytocin receptor in relation to behavioral patterns, including autism and other things. And their conclusion is these results suggest an association between variation in the oxytocin receptor, there were genetic changes, and human pair bonding and other social behaviors, indicating that the well-described influence of oxytocin on the affiliative behavior in voles can also be important for humans. Of course, the commercial people have gone to town. You can get liquid trust, and it'll do all sorts of things. You can get this one for autism, which is the oxytocin factor. You can get this one to make your love life better. You can get this one for romantic love. It will increase also fear reduction, trust, So, the commercial guys are not slow to get on this, but I guess none of this has really been satisfactorily proven. And then the media are just as bad. Oxytocin, the love hormone, could cure shyness. Telegraph, oxytocin polarizes men's opinions of their mothers. Telegraph again, love hormone, oxytocin helps soldiers like each other and hate the enemy. And the US News, oxytocin, the trust hormone, could become the new interrogation tool. Trust drug may cure social phobias in the BBC. And then this guy, oxytocin, the trust hormone, could it rebond our troubled world? Okay, so that's enough of oxytocin. I do think it's provocative, and I think it's really interesting. And there's a lot of neurochemistry to do and novel neuroregulators, which I think are going to be, revolutionize neuroscience. So let me talk briefly about cortisol. CRF in the brain, hypothalamus stimulates ACTH, and then cortisol production, which feeds back on the brain, and in stress situations has influences on the brain as well as on metabolism. Let's not spend too much time looking at that. This is a simpler picture that I've produced. So when you're stressed, CRF is produced, ACTH, and cortisol is produced by the adrenal gland. Cortisol has negative effects on reproduction, thyroid and growth hormone, and the immune system. As you know, it's used in transplant therapy. But it remobilizes through metabolism glucose and improves cognitive actions in the brain. This is acute stress. So how does this happen? Here's a stress. This is cortisol or other parameters going up. When the stress is removed, it immediately goes down. But you can continue to stress, and you either get continuous responses like this, and you get a chronic stress situation, which is represented by elevated, prolonged cortisol. Or you get a desensitization so that you now have a poor response to stress. Now, stress is perceived as being one of the ravages of our age, but I think our attitude to stress is partly responsible. Acute stress and cortisol production, in my view, is very beneficial. It's highly beneficial. It mobilizes glucose, it stimulates brain activity, suppresses superfluous activity like reproduction, and works in concert with adrenal. Well, if you're wanting to run away from a lion, I don't recommend sex, you know. So that's acute stress, and that's a good thing, and we should not worry about that. Chronic stress is bad, highly detrimental. You get a diabetic state with elevated glucose, muscle and bone breakdown, suppression of the immune system long-term makes you susceptible to infections, And you get depression and CNS effects as represented in Cushing's syndrome. So there's a schematic that kind of summarizes this. This is your performance. This is the pressure. As the pressure increases, you get increased cortisol, and you actually can do much better. And indeed, this is a sort of optimum for stress. But prolonged stress leads to decreasing performance. So I was interested in this kind of concept many years ago, and actually 30 years ago. I went to Tim Noakes and I said, we want to do a study, get us some marathon runners to see if overtraining is to do with this phenomenon of stress and cortisol production. And we got some really staggering results. Here's cortisol production in overtrained athletes. This is their ability to respond to stress. We stressed them with insulin. The ACTH is also low, as you'd expect, and growth hormone. After resting for 4 weeks, this response becomes normal. And then here, these are the comparators of the 42-kilometer, 92-kilometer marathon runners before and after of the marathon. So you see, they're not impaired at all. So the neuroendocrine system is a very, very sensitive monitor of physiological state and athletic state. And I think we have a big opportunity to explore new neuroendocrine aspects in relation to athletic performance. So the last thing that I'm dealing with is the neuroendocrinology of appetite, metabolism, and obesity. This is a highly complex, as the energy utilization and appetite are finely tuned by diverse neural inputs. I mean, I always used to think it's a remarkable thing that I could go for many, many years through my life eating exactly what I wanted and exercising exactly as I felt appropriate or inappropriate, and my weight didn't change at all. My weight has started to change. But for some people, this is a real dilemma. And this cartoon represents the immensity of this problem. At the waiting room, obesity is sitting right at the front door. Global warming, take a bow. Obesity is the biggest pandemic problem of the age. South Africa does not escape. We are up there with the rest of the world. We have up to 30% obesity, and morbid obesity is particularly high in South Africa. So how do we regulate appetite and metabolism? Well, it's quite complicated, but compared with the central nervous system, it's probably quite simple, and we know a lot about it. In the arcuate nucleus is a collection of cells that are orexigenic or anorexigenic, i.e. stimulating feeding or not stimulating feeding. But see how they're all connected up, and they talk to another place, the NTS. So it turns out this is not so simple to try and solve. These are the factors in the periphery, i.e. from the gut, from the stomach, from fat cells, from the pancreas, that feed into that area, and some of them are stimulatory and some of them are negative on these collections of cells. Now, the pharmaceutical industry has put huge effort into making PYY and GLP-1 agonists and antagonists and so on in an effort to make a huge amount of money, because with the obesity pandemic, there's tons of money to be made. None of these drugs work. And that's perhaps not surprising, because we have redundancy and feedback in a complex way up here in the arcuate nucleus. So if you take one of the things out of the system, or you put one in an inhibitor, it's unlikely you're going to achieve the job. So this is the outcome of this. If we look at the body mass index, look at the increase in diabetes related to obesity. In fact, the line of diabetes and obesity just follow each other perfectly. You could almost say that obesity is the singular biggest cause of diabetes. And then if you look at things like vascular and coronary incidences, coronary heart disease, they're also following the trend of being more, you're at greater risk if you are obese. And when it comes to chronic illness, I picked up these USA data. You'll see here there's a complete, a line which is really up at 48% of the population already is suffering from chronic, non-communicable diseases. And in front of these are diabetes, heart disease, and depression, which are all related quite strongly to obesity. So how are we going to deal with that? So I'd like to end by saying that all neuroendocrine systems the neuroendocrine system integrates inputs into regulation of most areas of body and brain function. The neuroendocrine system is the mediator of most dialogues between the environment, body, and the brain. And neuroendocrinology has become a major target for pharmaceutical intervention and has considerable potential now in what's called psychotherapy or psychoneuroendocrinology. There's a lot of excitement about that. I've only brushed on the edges of this vast galaxy. In fact, you know, it's just like nothing that I've done. And brushed on what is known and unknown in neuroendocrinology. But it is a fairly tractable field. We know the players, we've got the tools to investigate it, much more than some of the things we'd like to look at in the central nervous system, like depression and schizophrenia and so on. So I think that further exploration promises many more exciting discoveries and application in human and animal health. I want to finish by quotes from Aristotle. There are many. I recommend his book. You could probably get one from Amazon. You can, actually, because they are translations. The first is that it is a mark of an educated mind to be able to entertain a thought without accepting it. And the other is what we attribute to lots of other people, but he's the one who wrote this: the whole is more than the sum of the parts. And he recognized that in many of his animal studies. So I commend that as an institution and also as a country, these are two very important things that we should try and address. So thank you.